Hello, Bioscience 101 students. This is lecture 13. We're in the home stretch. 2 more lectures to go beyond this one. So today we're going to talk about fluorescent proteins, or mostly about GFP, green fluorescent protein. So here it is. This is a cartoon drawing of the structure of the protein. It has this interesting barrel shape where it's sort of hollow in the middle. It's short and stubby with a hole in the middle. And as proteins go, it's tiny. It's only 27 kilodaltons, if you remember your chemistry or whatever. That's a teeny tiny protein. So it's easy to attach it to other proteins that are way larger. This is just like a little bump. And as it turns out, it's super easy to attach it, and it doesn't seem to, so far, ever affect the function of proteins. I mean, you know, once in a while you attach it to the wrong spot, but you'll notice quickly. But there's just been a huge number of proteins where we're able to add it on to the end of the protein, and it's just this little extra bit of amino acids hanging out and fluorescing, and it doesn't seem to affect the cells. It's not toxic. It doesn't change the results when we compare it to known results. So it's been an amazing, amazing tool for biologists. Here's what we do with it. We transfect cells. That means we give it a vector, also known, often it's a plasmid. So it's a little circle of DNA if it's a plasmid. And that has the genetic information for just GFP, if that's all we're doing, or the gene of interest with GFP, the code for it at the end. So we're going to make a fluorescent version of whatever protein of interest it is that we're doing. And when you transfect cells, when you're basically genetically modifying them, you're not going to have 100% success rate. The company that sells you the reagent called lipofectamine that you add to cells, it's going to claim it's 70-80% transfection rate. In real life, you're like, you'll make do with 20, you'll be thrilled with 40% transfection rate. So in other words, if 40% of your cells glow green, you're thrilled. This is why most scopes that have fluorescence also have DIC on them, because you want to see all the other cells that aren't fluorescent, just to know, you know, what else is going on. In this picture, you see 2 beautiful cells that are fluorescent. They look healthy and happy. And then you see some other cells. The cell right above the fluorescent cell that's more on the right might be dying. It's got little blebs. It doesn't look very healthy. The cell that's super round attached to it, let's see if you can even see my cursor in here. Oh, can't. But you see one cell that's just balled up and super round, probably undergoing mitosis. And then if you notice on the corner there's a gigantic cell. That tells me these are cells grown in tissue culture, which is expected because, you know, I'm looking at a GFP transfection. But in cells grown in tissue culture, after a while, they just start getting all sorts of sizes and you get some extremely big cells. These are probably CHO cells, but I can't say that for sure. They look like, you know, a lot of cell lines actually look like this. All right, so that's what we do with GFP. This is the spectra. So this is the important thing to know about GFP. Of course, by now you're used to looking at spectras, right? So here's some excitation and emissions for other fluorescent proteins. Soon after GFP came along, of course, there were a whole lot of other proteins, including, well, YFP, which, as you already know from the last lecture, is a famously bad addition to the, possibilities, because it's practically indistinguishable from GFP, except when you do fancy things like FRET, exploiting the fact that it's really close, the spectra is really close to that of GFP. So here's what else we can do. Once we have more colors, we can label different parts of the cells. This is just showing you different colors and, you know, with different cell lines that have for instance, in C, you've got GFP tagged tubulin, and that's the microtubules in cells. Those cells actually look weird because the tubulin's surrounding the nucleus perfectly. But anyway, aside from that, in A, you've got the mitochondria, actin, which I've talked about before, the Golgi, vinculin, which is the little sort of feet of cells, histones, the F, and that are tagged with M cherry. The histones are the proteins that the DNA is wrapped around inside of your nucleus. So here are different colors just to see, look, different cell parts. Because it's a fluorescent protein, usually we're happy with one color, but these days people are getting fancy and even have doubly transfected cell lines, so even 2 colors. If I just showed you these pictures, there's no way you could tell me that they were a fluorescent protein as opposed to IHC, except for the fact that I'm telling you, like these could be fixed cells, in other words, but they're not. They're movies as per the metadata that was included, thank goodness. So here's one of the original sort of presentations from the Chen lab, the lab that gave us most of our fluorescent proteins. This is at a talk they gave. They just blew everyone away with this palette of different colors. And still, these are they're hugely in use. They're still just really, kind of the palette we pick from. But look at all these choices. And you already know that it's hard to multiplex with like 3 colors. So, I mean, it's just amazing how many choices we have. They're chemists, so they're just always playing around with the structure and finding new colors. This is just the fluorescent protein in a test tube. It's a composite picture. So these are different scopes, different images taken, and then, just thrown together, photoshopped together like this. So you can see the rainbow. And in fact, speaking of rainbows, here's an early notable use of fluorescent proteins, where most of us, I'm trying to go back, let's see. This palette came out pretty soon, within years of the first fluorescent protein, and most of us said, we'll pick 1 or 2. And the Lichtman lab, Jeff Lichtman at Harvard said, why stop? Let's use them all. And remember how I said that when you try to do the experiment, you're only able to genetically modify like 20% of your cells if you're lucky? Jeff Lichtman said, how's that a problem? That's good news to me. What most people are struggling with, I'm going to turn around and say, this is actually what I meant to do. And what he did was kind of crazy at the time, but it worked. He just threw a whole bunch of plasmids with different colors at neurons, at developing neurons in embryos of, I feel like I want to say chickens looking at this, but I'm not sure. But whatever model organism he was looking, he had a way to genetically modify the neurons. And he just threw the fluorescent protein into the brain as it was developing, and used statistics, basically said, I'm going to throw low doses of a variety of colors, and just the colors, not attached to any protein. And each cell will take up maybe 1 or 2 colors, and we'll have a unique color in that cell, and we'll just paint the brain. And I don't care what color ends up in what neuron. I just care that neurons end up with different colors. And based on the fact that this is going to work maybe 5% of the time, the chances of 2 neurons getting the same color and being right next to each other are kind of low. And he was right. You get these beautiful, he called it brain rainbows, where they're able to trace neurons throughout the parts of the brain using this technique, because otherwise all the neurons look the same. They have these long projections called axons and dendrites. And if you're trying to map the brain, you just see a lot of wires basically. But how do I know which wire belongs to which neuron? Where is it traveling from to in the brain? Unless you paint it and make every wire a slightly different color. This technique is actually, it sounds so easy, it's actually kind of hard to do, but they're able to do it and they get some good data out of it. Um, there's been fun with GFP. Early days in the GFP revolution, an artist made this glowing bunny, as you can see. Scientists were still debating, should we put this in actual animals, or are we just gonna, like maybe embryos that we test, but what are the limits? And this artist said, hey, I'm just gonna put it in a bunny and grow the bunny to be a full live happy bunny. And I remember a scientist going, wait, what? Like, don't you have to run that in front of a variety of commissions? And no, the answer was no. This person was an artist. They weren't regulated. So anyway, this little glowing bunny is quite famous. He lived quite healthy. It's a white bunny in real life, but this is under a dark light, basically. Yeah, it would fluoresce the whole bunny. And then these are some other pictures like showing twin pigs. One has the fluorescing yellow nose, the other one doesn't. The kittens, they did get protocols too, and they fluoresce under the right light. But again, all these animals seem to be fine. Like we haven't been able to observe any sort of detrimental thing. These are just more fun pictures of scary looking insects. They're again, separate insects kind of photoshopped together on the right. But there's websites, I hope this is still up. There's this entrepreneur somewhere in Asia was trying to sell GFP fish. I think you can still buy them actually. So they're glow in the dark kind of fish. And this is just a pretty picture of neurons with rainbow. And notice the names of the fluorescent protein. So green fluorescent protein is named that because it's green and it fluoresces and it's a protein. And soon after we got yellow fluorescent protein. And then E stands for enhanced. In other words, it's not the original. There's a little bit of genetic and chemical wizardry that makes it brighter and more stable. And everybody uses EGFP. So EGFP is like its formal name, but we just call it GFP when we're in a hurry, which is almost always. M stands for monomer because there's some that dimerize, which means their two proteins stick together. And so they started with colors. This mostly came out of the Chen lab, and then as you'll see, some other labs started making them. And and after they sort of picked a few colors, and then they just started going with fruits. Which I get a real kick out of. So orange, raspberry, strawberry, tangerine, cherry. Cherry is the one that is widely used. It was the first, well, not the first red, but and this is a guide to choosing them if you want to look at it. It's pretty fun and informative. So there's a lot of fluorescent proteins. Some of them are used constantly all over the world by everybody all the time. And others are, used once or twice. And it just depends on not just the color, but also what transfection level, the photostability, all these things about fluorophores that we know. So the Chan Lab sort of pumps them out and is like, go and use them. And then people, figure it out. And some are more useful than others. But here is an interesting use of one. And oh my goodness, I can't pause the computer. I'm going to go to let my cat out. I don't know if the howling is being captured on tape, but I'll be right back. So just a quick 10-second pause. Okay, so here is another use of, why is it so important that we have the ability to label life parts of our specimen. So here's another example of a good use of a fluorescent protein where they're coloring the HIV virus basically and watching it, kind of scary, snake from cell to cell without ever even just inside of a cell into inside of another cell, which is how it you know, evade the immune system. Here's an example of a use in a paper where they're just labeling the cells that are dividing as green and the cells that are just hanging out, chilling in interphase as red. So just fun uses and some cool pictures to look at. You can, as you saw by the artist with his bunny, but scientists have also since then done this in animals. You can make an animal fluoresce or parts of an animal fluoresce. And so often we're injecting tumors into mice that they don't have an immune system, they don't have hair, and we use them to study how tumors grow. And you can actually put a whole mouse on a microscope, and that's called in vivo imaging. It's got obviously a big working distance. If the mouse, there's a lot of rules and regulations about the mouse is basically under anesthesia and it's got oxygen. It's basically an operating table for a mouse on a microscope. And anyway, you can get tons of info about tumors, obviously, by labeling them and labeling different parts of them with fluorescent proteins and seeing how it changes. All right. So in 2008, the Nobel Prize in Chemistry was awarded for the discovery of green fluorescent protein. And it's interesting as I sit here and give this lecture or record it in 2021, because that's when we started the merit microscopy program. And it was so cutting edge at the time. It was so recent. And I was so excited to tell the students like, hey, this thing just got the Nobel Prize, which by the way, was awarded really fast because it's often 15 years later or 20 years later. And this was just, it wasn't that much later by those standards. So it's talking about the Nobel Prize is actually a good way to describe what GFP is and you know, how it was discovered and so on. So this is a naturally occurring protein. It's something, well, of course we've modified it, but for the most part, this is something that is found in jellyfish. So Aquaria victoria is also known as a crystal jelly or A victoria. And yes, we can find it off of our coast, and you can find it in the Atlantic and the Mediterranean. Is that even spelled right? But anyway, it's found all over the world, this particular species, and there it is. Monterey Aquarium has a whole display of them. They're pretty small, well, not tiny, they're 5 centimeters or 5 to 10 centimeters in diameter. So, you know, smallish, but you can see individual ones yourself. And they look like that basically. And what is going on is that they create their own light. Okay, so they have a protein known as aquarin. And if there's calcium around, which there is because they're in ocean water, so there's calcium, they produce their own light because it's deep and dark out there. So there's a lot of critters in the ocean that create their own light. It's a whole different world. And they create blue light. And oh, as you know, blue light excites GFP, right? And so their own blue light hits the GFP that they also have actually attached to Aquarin, and that fluoresces green light. So they're using chemistry, really cool chemistry, to create their own blue light. And then they've got a protein that fluoresces, that is GFP. And here's some more really cool photos of them because I couldn't resist, because look how cool they are, right? I mean, they're just beautiful. And so this is called bioluminescence because they are producing light out of nowhere. And you see it with a lot of organisms when you go out in the ocean, and sometimes in tides at night when the sea kind of shimmers or the waves glow. That's possibly these guys, but also a lot of other organisms that exhibit what is known as bioluminescence. So biology making light. So this was known, but it wasn't known how this happened. And so it took mostly the work of the 3 men who won the Nobel Prize. Osama Shimomura is, he was working at Woods Hole, which is this great place for biology, marine biology, but all sorts of discoveries come out of it. It's up in Massachusetts. It's actually where I went to learn microscopy when I was setting up this program. So he was born in Japan and went to Princeton and worked did his work at Woods Hole. And he discovered aquarin, and he's holding a vial of, aquarin plus GFP. So these things that he called squeeze-its, where he would squeeze the jellyfish. Notice it's on the coast. So Woods Hole is, so it's actually marine biolabs in Woods Hole, Massachusetts. And it's a research lab funded by taxpayer money. Thank you, everybody. Right on the coast. So they go out and get stuff, including jellyfish, and bring it back to labs and study it. The second person on the Nobel Prize was Marty Chalfie, who is actually as jolly as he seems in these. Like, it's hard to find a photo where he's not smiling. I've heard him in talks. He's just a very upbeat person. And he is the person who really did the groundbreaking work of using GFP in living cells. And interestingly enough, he went on to do other things for his career. And I love this picture of him at a microscope because, so he published the paper where, GFP was announced to the world. And he actually, and then, won a Nobel Prize, revolutionized all of biology forevermore, started, basically led to us starting the merit microscopy program, because there's such a demand for fluorescence microscopy, and it's evolving, and there's so much going on with it after the GFP revolution. He didn't own a microscope. He actually had the sales reps loan him a demo. So when they're trying to sell you something, they'll loan you a demoscope for a week or two. And he did all his experiments on that demoscope. So I think that is so clever. And, scientists hustle for their stuff. He wasn't famous or rich at the time. He just had a crazy idea that worked out well and changed the world. So, and he's at Columbia. And this is Roger Chen, who he passed away, unfortunately, quite young, relatively speaking. But he was as serious as his photo, but a delightful person, a really fair person, a brilliant scientist. My lab kind of collaborated with his lab. So I heard a lot about him. He was at UC San Diego. And this is some bacteria that somebody in the lab made, using different fluorescent proteins. Okay, so here's a quick history of it. So as I said, so Shimomura got was rightly on the Nobel Prize because he is the one who figured out what was up with the jellyfish, or he even decided to study these jellyfish and went out and got thousands and thousands and thousands and thousands and thousands of them. And he would literally squeeze them through cheesecloth and get jellyfish juice from the poor little dead jellyfish. And he called these squeeze-its. And he started this work in the 60s. So I say it's relatively quick to, a Nobel in 2008, because it was only 10 years really. But of the sort of modern stuff, but it goes all the way back, like a lot of science does, to just doing things by hand out of curiosity and going, jellyfish, why are they lighting up? I want to know how they do it. And that's what Shimomura did. And he just kept studying it. And he, identified aquarin, and then he found he just broke up what he had into pieces, and he found a piece that he realized was a protein, called it GFP, and went on to study it, and what's its structure, what's it doing, how is this all working? And then Doug Prasher was another scientist, collaborated with him, and he took it to so Shimomura was there doing biochemistry in the lab with the actual jellyfish. Prasher was like, this is cool what you're doing. You know what? I want to try to put it in a vector, in a plasmid, basically. So when I say cloned and expressed aquarin and then GFP, that's what he did. So they laid the groundwork. And then in 93, in the early 90s, basically Chalfie, Prasher, and Chen are all working on this. And in 90, and Chen starts to understand the structure, because remember, he's actually a chemist, although he pretty much made a lot of stuff even before this. He was making fluorescent probes for imaging for biologists. So we think of him as a biologist who knows a lot of chemistry. So then this bombshell paper comes out in 94. I still literally can remember where I was when I picked up the copy of Science magazine. I looked at the cover and I went, what? And then I ran around the lab and talked to everybody. And then my lab I was the only one into imaging in my lab. So I ran out in the hallway. They were like, we don't get it. And I ran out in the hallway and started going to other labs who use scopes. And everybody was like, what is going on? Oh my God, we can do fluorescence in live organisms. This is crazy. So you can tell I'm still excited all these years later. I'm like, oh, that paper. Okay, so Chalfie and Prasher published this paper that I'll talk about in a little more. And they did it in E. coli, which would have been enough. We would have been like thrilled, like, oh my God, you got something that doesn't fluoresce and it's alive. Oh my God, like that's amazing. Because at that point, remember, all fluorescence, we were pretty much dealing with fixed stuff, except for actually a few dyes that Chen had, but they were difficult and tricky and so on. In fact, I think those years, I was working on one of his dyes that didn't work out, actually. Lost a lot of time over that one, but oh well. Anyway, so they did an E. coli, which is pretty cool, but bacteria are very different than mammalian cells, and or animal cells, let's say. They also did it in C. elegans, which is a worm, which is an animal, not a mammal, but an animal. And so it was like, what a feast. There was Shinya Inouye is this amazing imaging scientist who did a lot of landmark work on imaging time lapse, which is movies, basically. It has this amazing lab, and he also published around then. But in science, if you're the second one, then you're not the one everyone remembers. And then, so of course, the minute the science paper came out, was Marty Chalfie. Everybody's like, I got to do this, and then. They realized it's hard, but in just 2 years, the Chen Lab had already created the better version of GFP, and then a bunch of colors, basically. And it took what felt like forever at the time, to be honest, to get a red. A red was just like not a color that we saw that wasn't natural. It wasn't in nature. It wasn't in all these marine organisms. They weren't fluorescing red. And remember, autofluorescence tends to be blue and green. And so were we going to find, a nature created fluorescent red? And people were synthesizing them. Somebody in my lab was trying to do it. Everybody was on this. They're like, oh my gosh, let's find a red. And actually a different lab, not the Chen lab, got the first red by looking at corals, because they happened to know enough marine biology to know that corals do fluoresce red, actually, while the rest of us, we were like, what's a coral? And corals turned out to be a really good source of various fluorescent proteins. And eventually the Chen lab did get to M. cherry, which again, people use because it's really good. But DS red was the first one. I look at this timeline as I teach, and to me, it seemed like it was 10 years, but it was only like 3 years to the red. But it's just everybody's attention every day. Is all we were thinking about. We were obsessed. So every minute of every day, it was like, what can we do? What what fluorescent proteins are out there? So here's what Doug Prasher did. Just a little thing. He really worked out the molecular biology. So the genetically modified part, where you put it into, how do you get the DNA into a vector that's going to go into cells and all of that. So it was nice and ready for Marty Chalfie to used in an interesting way. And arguably he should have been on the Nobel Prize because, but really it was 4 people: Shimamura, Prasher, Chalfie, and Chen, who really made this happen. And in these cases, usually it's people in the lab, but it was in this case, it was these actual people who were doing the actual work. Um, and there's the Nobel acceptance speech is really fun and interesting. But even though it was what, like 10 years later, they got the Nobel. At that point, Doug Prasher was no longer in science, and they can only have 3 people on the Nobel Prize, and they have to. So it's just political, basically. And the cool thing, though, is Roger Chen, like when he got the Nobel, he was just in the lab busy doing stuff, and we got the Nobel. He was like, "Wait, what happened to Doug Prasher? Where is he?" And he actually, when he found out he wasn't in science anymore, Chen like basically said, "Come to join my lab, and just you can be a scientist in my lab," which is, I think, really cool. So better than pretending he never existed. Like he was very generous. Chen was. Okay, so here is, well, that's the story, really. Since then, oh, how come? How did we? Okay, my cursor is going in all kinds of bad directions. Come on. Let's go back. Okay. So here's the original paper. So we're going to end this with a look at the original paper because it's a landmark paper in science. It's one of the most important papers ever. And the good news is it's also super simple and short. I highly recommend looking it up. I'll try to remember to post it to our Canvas shell. And it's not just short, it's really clear, and it's a great way to look at an actual original research paper and understand it. You should be able to understand it at this point. So the title is very short and very simple. It's green fluorescent protein, so GFP, as a marker for gene expression. Gene expression means you're using your genes. And Marty Chalfie, other people I should know but don't, and then Doug Prasher. And Columbia University. Science is one of the most prestigious journal, really. Science and Nature are the 2 most prestigious journals to publish a paper in. And so that's the title, that's who did the work, that's where they did it. And then here's the abstract, which is a summary of the paper. And this abstract is so beautifully boiled down and brief that you can even look at it, and let's look at it together and just break it apart. So a cDNA, complementary DNA, that just means the DNA for it. For the Equaria victoria, that's the jellyfish. Green fluorescent protein, that's the protein that's green and fluoresces, produces a fluorescent product. So it fluoresces when expressed in, so when you add the DNA, you transfect the DNA, and then it's inside of, we're going to see what in a second. And then it could just sit there and not be used. Expressed means the cell actually uses it, because not all DNA is used all the time. In fact, most isn't. So the cell does actually take this DNA and turn it into an RNA, and then make proteins out of those genes that you gave it. You're giving it the genetic info, it actually uses it. So expressed means it's used, it's turned into a protein. And then of course, once you have that protein, you can see it under a scope. So where is it expressed? Prokaryotes, so bacteria, E. coli, yes, that E. coli, the one that's in our guts and used in labs and so on. Prokaryotes are types of organisms that don't have a true nucleus. And so this is like reaching across lots of kingdoms and species and so on. And eukaryotes, in other words, things that have a nucleus, including, for instance, us, and in this case, worms. Cells. So in the cells of bacteria, in the cells of C. elegans, C. elegans is a little elegant, it really means elegant, worm that we use a lot in lab, is really important in the soil. It's this tiny, super cute worm. You're going to see it in a moment. And then here, this is so understated, but it says, because exogenous substrates and cofactors are not required for this fluorescence, In other words, there were really difficult ways to make things not really fluoresce, but more phosphoresce. But there were ways to sort of light up things before, but they were ridiculously hard so as to be pretty much impossible and impractical. And you had to add a lot of things from outside. So exogenous means from outside. You had to add all this other stuff to it. And it just almost never worked. It was so hard and it just didn't work. So for those of us in the field, we're reading this sentence, we're going, you don't have to add anything else. Oh my God. All you have to do is get the cell to take up the DNA and then use it, express it. That's easy. We knew how to do that at the time. So then so it's saying, well, because, this one's easy. Yeah, I don't have to do anything extra. I just throw the DNA at the cell and then I get fluorescing bacteria and, oh my God, fluorescing animals. Um, and then then there's a sentence where it understates the revolution. It says GFP expression. So, having GFP in in cells can be used to monitor gene expression. In other words, study what's going on with genes. How do genes get used and turned on and not used and so on. That was a big field. It still is. And to me, this is the bomb drop. Protein localization, like where are proteins? Where are they? Because where they are is everything about what they're doing. So where are they? In what parts of the cell? Who are they next to, et cetera. So you can use, this is them dreaming and saying, hey, we did this thing and we think y'all might want to use it. Anybody interested in studying where proteins are? Which is like all of us, anybody interested in studying how proteins are made and which DNA is chosen when and how? That's gene expression, which is like everyone else. Like we think this might be useful in these fields that are like everything. And then the real bombshell, the real like sort of confetti goes off when it says in living organisms. It's like, what? Blow my mind. Like we're studying they're convoluted ways with fixed cells. Like we're doing all these incredibly like difficult parallel experiments where I start something and then I fix it at time point 0 and I fix it at different set at time point 30 and time point 50 minutes, and I'm comparing things and it's so much work and it's not really good because I'm not really looking at the same cells. You just told me I could do all of that in a living organism and just make a movie. Oh my gosh. Okay. Yeah, I still react to it this way. I'm still excited all these years later. That's how you know you found your field. So this was the cover of Science that I still have my copy of. And I love showing this because, so February 11th, 1994, back in the day, before, I think this is before the internet and Google and all that. Anyway, like we got physical copies. They landed in our labs. We looked at them, not usually with this much excitement. And I love showing you guys this picture because getting the cover of Science is a huge accomplishment. And this is a worm, so you can see its two little eyes. Oh, this pointer does not work, but you can see sort of there's the two dots under the word science. That's its head, and those are its eyes. And what's actually labeled are certain neurons in it. And then there's just sort of a background glow in the whole worm. And the worm sort of is this, nice, cute shape. It's just curled up on itself a little bit so that it fits in the picture frame perfectly. So nice of the worm to do that. I'm sure he only had those scopes for like 2 weeks. So I'm sure he was imaging like crazy. And, this is the one that he picked to be the poster child, literally the cover image. And what I love about this is not only the sort of historical significance, and again, looking at this, do you know it's live? No, because you were told it was live, and we, saw that. And we were actually, I think my first reaction is, why is this is not that great a picture. And then opening and going, wait, it's live? It's live? What? And that's why I like showing it to you, because if this was, like if you were in lab and you were taking this picture on our scopes today, I would be like Are you kidding me? You can't turn that in. Focus. This is out of focus. This is blurry. Signal to noise ratio, not very good. This is so dim. Come on. Oh, look, the eyes, they're saturated. You can do better. And you can do better if you're looking at GFP worms or pretty much any fluorescence on our scopes today. But it doesn't take perfection to rock the world and, start a revolution and change everything. It just takes something really profound. And at the time, that was as good as the scopes and the, remember, he was trying this on free scopes. So as good as the scopes and the cameras and everything got. And in fact, this poor thing was alive and wiggling. So it's lucky it's not even blurrier. And so it didn't matter that it's blurry or saturated when it's this important and profound. And it was kind of cutting edge technology. It wasn't kind of, it was cutting edge technology at the time. And this is the figure legend for that. So you can see that it's the neurons. I'll let you read this. And in the paper, the original paper, there's also this figure of comparison of bacteria, fluorescing on one hand on the right side and not fluorescing on the other side. With a handheld long wave UV source. It's much like those pet stores sell these stink finders, or just UV lamp, basically low tech, but still revolutionary. And I think that's it. Oh, this is Chen Lab. They show this at a lot of talks. It's a bacterial plate they made with, lots of different fluorescent proteins. So anyway, so basically lots of work over decades, but really there was Shimomura doing his thing for a while. He moved on to other things, and then, kind of Doug Prasher figuring out some stuff. And then it just blew up in the 90s with the Chalfie paper, and which Prasher was on. And but Chalfie really was driving. He gave credit to Prasher because Prasher had made the plasmids, and Prasher literally just handed him a vial and sort of said, have fun. And then Chalfie got some scopes and some people and some bacteria and he was already working on worms, so he knew how to use worms. And he said, I have this crazy idea that I'm going to try to make a worm fluorescent the same way that a jellyfish does. And it worked, and it revolutionized the world along with Chen Lab's input, and lots of people have been using this for all sorts of reasons and all sorts of discoveries.